Hey, hello, and welcome all of you. Well, today we will talk about an interesting topic called DNA fingerprinting. My friends, DNA fingerprinting is also known as DNA profiling. Well, in this video, we will discuss the process and applications. An in detail, step by step video will be produced or released later. Well, as of now, let me start by introducing where it can be applied. Well, all throughout the world, rarely but surely, there are cases of parental dispute. All right. So people do not have an idea who the real father is, and sometimes whether the child belongs to both the parents or not, due to the child getting exchanged in the hospital when it was just born with other children. And there are cases like, as you can see in this movie, Amar Akbar Anthony, or these three people when they were children, they get lost during the Kumbh Mela, right? And when they grow up, they meet. So in all such cases, DNA fingerprinting can prove to be an effective tool. Well, there are cases of sexual assault or rapes where the suspect can be verified with the help of DNA fingerprinting. There are cases where sexual assault may be followed by murder, and it gets more complicated. In such cases, since the victim has not lived to tell the tale, the suspects might not even be discovered. So, rape is a bad crime, and when it is followed by murder, it gets even worse and more complicated. The victim is not alive to tell the tale. All such complicated cases do resort to DNA fingerprinting. It all starts by collecting samples or collecting the evidence. It is the forensics team that collects samples from the crime scene. Samples like hair follicle, skin, bone, even saliva, which may be present on the rim of such cups present at the crime scene. Many a times, even blood stains of sperms left on linen, on sofa cushions, bed sheets. So, all these samples or evidences are first collected and employed in the technique of DNA fingerprinting. The basis of the technique lies in our genome. So, as you are very well aware, that human genome sizes up to 3 billion base pairs. But let me tell you that we, the people, are around 99.9% same. It also means that we are only 0.1% different from each other. Well, percentage wise, it might sound a smaller number, but when you calculate 0.1% of 3 billion base pairs is around 3 million base pairs. During a crime investigation, if you try to identify people based on 3 million base pairs, it is going to be a daunting task, and it will also be expensive. So, isn't there a way which is quick and easy? Yes, my dear friends. It is DNA fingerprinting, which can be employed to profile individual suspects and compare in between them within no time. So to start with, on the left hand side here, you can see a DNA molecule which has undergone some kind of modification, which we term as mutation. So DNA molecule which has undergone changes in its base sequence is called as a modified DNA, or you can also call it a mutant DNA. So if there is a mutation in the DNA of an individual which has not affected its reproductive ability, this mutation might get passed on to the next generation. These people have born children, and then those children, in turn, will bear their own children, which will marry other people. And over a lot of generations, the mutant gene would spread across the population. If at all, this mutant DNA was part of a functional gene, a fragment of DNA that is involved in protein production. If such a gene, which is involved in protein synthesis, undergoes mutation, it will also produce a phenotype. So as you can see on the left-hand side here, there is a pair of homologous chromosomes. They bear genes for the same characters at the same location. That's the reason why they are called homologous chromosomes. And from every homologous pair, one is what we receive via the sperm from our male parent, that is from our dad, and the other one we receive from our moms. So as you can see, this particular homologous pair has genes for eye color. On the right, it has gene for brown eye color, and on the left, it's for blue eye color. Let's say one of these two is an original gene, the brown eye color gene, let's call it for now as the original gene which initially evolved. But later on, due to some kind of mutation or some kind of change in the base sequence, another gene came into existence which would give rise to blue eye color. A change in the eye color does not affect eyesight, and it definitely does not affect the reproductive ability of an individual, as does a change in the skin color, or there are many other aspects like genes for free earlobes or attached earlobes. Right? There are so many variant genes out there in the population which do not affect the reproductive ability of an individual. And such variant genes, such alleles, get transferred from one generation to another and over a few generations become part of the population. When such variants or alleles or deviations from the original gene become part of the population and their frequency goes above 0.01, that is, their number or the individuals bearing that alternate gene, when such individuals are more than 1 in 100, at that time we are allowed to call such variant or mutant fragments of DNA as allelic sequence variation. We can recognize such mutations as an allele. Officially, we can call it as an allele. Why? Because it is now spread across the population in good enough numbers. We can also call it as DNA polymorphism. Poly means many and morph means form. So my dear friends, allelic sequence variation or DNA polymorphism is nothing but more than one inheritable variant or allele due to mutation in the original allele. When this mutation takes place at the same locus more than one time, and if that variant or allele spreads across the population over a lot of generations and its frequency gets higher than 0.01, we are allowed to recognize it as an alternative form or an allele. It is also called as allelic sequence variation. My dear friends, such mutations can also occur in the region in between genes, not within the gene. In the previous slide, we discussed a mutation within the gene. But now, let's talk about this intergenic region, which we also call as junk. Since it is not involved in protein synthesis, we thought it is useless. It does have base sequences, but it does not code for any protein. We call it junk. But now we know that this junk region has a lot of role to play in gene regulation, but as of now, that's not the topic. Mutations that take place in the junk region. Let's talk about these mutations that take place in the junk region. Let me tell you that probability of such a mutation being observed in non-coding or junk DNA is very high. You know why? Because if mutations or variations take place in the gene, they obviously affect the phenotype, and when they affect the phenotype, sometimes it may end up the individuals losing their reproductive ability. Sometimes it may even threaten the existence of such an individual. So any mutation that is caused in the gene has lesser chances to get carried forward to the next generation as compared to the mutations that have taken place in the junk region. So therefore the statement says that probability of such a variation being observed in non-coding or junk region is high, because variations or mutations that have occurred in the junk region may not have a direct impact or an immediate impact or affect the reproductive ability of an individual. So such mutations which have occurred in the junk region accumulate with time and get passed on from generation to generation. And they are the ones which form overall, even the gene mutations and the junk mutations are the ones which form the basis of variability or polymorphism. This polymorphism that takes place either in the gene or in junk may may range from a single nucleotide change to very large scale changes that may take place. And the effect totally depends on how it goes ahead, which protein it affects, or maybe it does not affect anything, right? Now, let's take this as an example. This is the junk region between two genes on a particular chromosome. And as you can see that this particular repetitive DNA, why am I calling it repetitive? There's a sequence here, GATC, So this particular sequence is just repeating over and over and over within the same chromosome. How is this possible? This is possible due to a copy paste mechanism which ends up modifying the DNA molecule. So this chromosome, whichever number of chromosome it is, chromosome number 3, it could be chromosome number 5 or 10 or 8, whichever it is, it has this particular repetitive DNA, and I'm calling it repetitive because it is repeating itself over and over again. And not just that, it is repeating itself in a tandem sequence. That is, the repeats are placed next to each other, one after the other. Such repeats are to be called as tandem repeats. So as you can see, this is a pair of chromosomes that I've shown here. One comes from the male parent, the other comes from the female parent. And as you can see that the number of times that this particular sequence repeats male parent is 4, and the number of times it repeats from the chromosome in the chromosome that one has received from the mother, it has repeated only twice. So within an individual also, you will find the same repetitive sequence occurring twice, because every sexually reproduced individual has received chromosomes from both the parents. So whoever this individual is has received a chromosome from his dad, on which this particular sequence repeats 4 times, and on the chromosome which he or she has received from mom, this particular sequence has repeated itself twice. The number of times that a particular sequence repeats itself within an individual may or may not vary. So as of now, let's just keep it to tandem repeats. Okay. Why we call them as tandem repeats? Because those sequences repeat in continuity. They are one after the other. They get copied and then they get pasted. And every individual, since they have a pair of every chromosome, they will have the same repetitive DNA twice. But the number of times that it will repeat on paternal and maternal chromosomes, it might be the same or it might be a different number. So let's take another chromosome down here. As you can see here, there's a different sequence this time. It is CTTT ATAA, and this sequence has repeated thrice on the chromosome which this individual has received from his father. The same sequence has repeated 1, 2, 3, 4, 5, 6 times on the chromosome which he or she has received from his mother. Up here and down here, these are two different repetitive DNA sequences. Each of these repetitive DNA sequences is present on both the chromosomes of homologous pairs, and the number of times that they repeat on a particular chromosome may vary from the number of times it will repeat on its homologue. This diagram, on which we have shown 13 different repetitive DNA sequences which were identified probably by the American government for being used in case of DNA fingerprinting. These are 13 different repetitive DNA sequences. On this slide, up and down here, you can see only 2 of them. I hope you have understood that when you try to talk about 13 different repetitive DNA sequences, you will actually be dealing with 26 chromosomes, because for every chromosome you have a pair. So each repetitive DNA, let's say this one which is present on chromosome number 2. Now you do not have a chromosome number 2 present in a single dose. You have a pair of chromosome number 2. That's the reason why you will have the same repetitive DNA present here, the one that they've called as TPOX, in a pair. So this repetitive DNA will be present on the paternal as well as on the maternal chromosome. Chromosome number 3, 4, 5, and all these chromosomes that you are looking at, they all exist in pairs because we are diploid in individuals reproduced by sexual reproduction. So we have chromosomes coming from both the parents. That's the reason why every repetitive DNA will be present on both the chromosomes of the homologous pair. Let me give you an example, a very specific example. Let's take 3 individuals here and let's talk about chromosome number 3. I hope you remember chromosome number 1, 2, and 3. This one. Let's assume that this particular sequence D3S1358 is the one that I have taken here. As you can see, in individual 1, it is repeating itself around 8 times, but on the maternal chromosome, it is repeated only twice. And that's in individual 1. Whereas in individual 2, the same sequence is present on chromosome number 3 at the same location, but the number of times that the sequence repeats itself in individual 2 differs from the number of times the same sequence has repeated itself in individual 1 or any other individual in the world. One of sequences may be repeating same number of times in two different individuals, but not 13 sequences. So let me take one more chromosome. Let me also tell you here that since number of times that a particular sequence repeats in different individuals varies, so a particular repetitive DNA repeats for different number of times in different individuals. Because of that, we can call it variable number of tandem repeats. What are tandem repeats? A particular sequence of DNA which repeats itself over and over and over and over and over on a particular chromosome in a tandem arrangement where these repeats are placed next to each other. That's why we call them as tandem repeats. Why do we use the word variable number of tandem repeats? Because these repetitive DNA sequences repeat for different number of times in different individuals. Like you can say that this particular sequence on chromosome number 3 has repeated itself 8 times on the paternal chromosome of individual 1 and twice on the maternal chromosome, whereas in individual 2 the number of repeats is 3 and 5. In individual 3 the number of repeats is 6 and 4. So the number of times that these sequences repeat themselves in different individuals varies. So since it varies between individuals, we call it variable number of tandem repeats. Let me show you another chromosome. This is chromosome number 5. Can you see? Now this repetitive DNA, since it is a different chromosome altogether, it's a different sequence. I hope you remember those 13 sequences. So now we are done discussing chromosome number 3 and we are just moved to chromosome number 5, and after this I will also show you the repetitive DNA on chromosome number 8. So here we have chromosome number 5 and chromosome number 8. Can you see that all these chromosomes, they have their own repetitive sequences present in pairs, one on the paternal chromosome and one on the maternal chromosome. But the number of times that they repeat in different individuals or across different individuals, that varies. Can you see? This particular sequence on chromosome number 5 in individual 1 repeats 4 times on the paternal chromosome, and once it's not copied, it's just present once on the maternal chromosome. Whereas in individual 2, the number of times is 6 and 4. Individual 3, the number of times is 1 and 6. Now, you would feel like saying that in individual 3, this particular sequence is repeated only once, and the same you will find in individual 1. So that's the reason why I told you earlier that one of the fragments may have the same type and length in different individuals, but the entire repetitive DNA that we have, like if we are considering 13 repetitive sequences, which means we will have 26 such DNA sequences, because for each repetitive DNA we have two sequences. So for 13 repetitive DNA sequences we have 26 fragments which can be obtained. I'll tell you what fragment means in a while. So for every individual, you will get 26 such fragments if you just cut out these repetitive DNA sequences from the chromosome where they belong to, and you will get 26 fragments of individual 1, 26 fragments from individual 2, 26 fragments from individual 3. One of or couple of fragments of couple of individuals might match up, like this one between individual 3 and individual 1. Also, individual 1 matches with individual 2 when it comes to the same sequence, but since it is repeated 4 times in individual 1 and 4 times in individual 2. So if you obtain fragments, this matches this one matches this, and even here this particular fragment matches this. So now let's break them down into fragments so that we can study this further ahead and make it easier to understand. So what I've done is I have removed the bulk DNA and I have narrowed everything down to fragments which contain only the repetitive sequence. How did I do that? By cutting these chromosomes or DNA molecules using restriction enzymes. That's the reason why now these fragments are to be called as restriction fragments. You will study restriction fragments in the chapter biotechnology. DNA can be cut with the help of enzymes called restriction enzymes, and we can obtain such fragments. So here on the left hand side, you can see all the restriction fragments which are produced by cutting or by isolating the repetitive sequences from the bulk genome. These are the restriction fragments of individual 1, then individual 2, and individual 3. Can you see that the length of these fragments in different individuals varies? That's what we call as restriction fragment length polymorphism. I have already explained you VNTR, variable number of tandem repeats. That's the reason why you have RFLP. Now, what do I mean by that? So, since the number of tandem repeats in different individuals varies, any particular repeat sequence will vary for different number of times in different individuals. Because of that, the fragments or restriction fragments which will be generated as a result will be of different lengths. So, there will be a polymorphism. Poly means many and morph means form. So all these fragments across individuals will be of different length because the repetitive DNA varies for different number of times in different individuals. A couple of fragments might be of the same type and length, but it is of no chance that two individuals will have all 26 fragments of the same type unless they are identical twins. Let's pool all these fragments of different individuals together. So I have assembled, let's say, all the fragments that I had obtained by cutting and isolating the tandem repeats of individual 1, and I have pooled them together, individual 2 and 3 as well. Then we need to load these samples on gel to carry out electrophoresis. We apply charge, negative charge on one side and positive charge on the other side. We load the sample towards the negative electrode and then we turn on the current. DNA, being negatively charged itself, will run towards the positive electrode. When DNA fragments run, the smallest will run the fastest and the furthest. The entire gel electrophoresis process I will cover in another video. But as of now, please observe that the smallest of the fragments have reached the farthest and the largest of the fragments are left behind. They stay behind. So as you can see, I have unique band patterns of each of these individuals and they do not match with each other. Now let me tell you, if I take two different samples, let's say of individual 2, one from his hair follicle, another from his saliva or sperm or blood, both the samples will match because they come from the same individual. So in short, what I'm trying to say is that two different samples or many samples from the same individual will produce the same band pattern. The band pattern of one individual will never match the band pattern of another individual. So this is how, by spotting unique differences from the junk DNA of different individuals, we can solve difficult criminal cases. But again, like I said, criminal cases or crime scenes are much more complicated than what we think. So let's move further ahead. Can you see the band pattern? Let me show you an actual image of such a band pattern. This is how the samples are run in individual lanes. Can you see the columns which have DNA bands in them? So these are individual, each of these band patterns, every single band from top to bottom represents one individual. So when you are running multiple samples in different lanes, it means you are creating multiple DNA profiles. All you have to do is match it with the crime scene. Let's practice some exercise. What do we have here? On the left hand side, as you can see, there is a sexual assault case, which actually means that the victim is alive and she is claiming that she has been sexually assaulted or raped. So here in this chart, as you can see, we have the sperm DNA, we have female cells, and what's this thing called ladder? Ladder is sequence of artificially synthesized DNA molecules which are of known size. Probably it comes along with the kit. So these DNA molecules which form the ladder, since they are of the known size, they can be used as a reference frame to determine the size of unknown samples. All right. So it's just to determine the size of the unknown samples. So ladder is not part of the evidence. Ladder helps us to determine size of the unknown samples. What all samples do we have here? Apart from ladder, we have sample from the boyfriend. We have suspect 1, suspect 2, victim. Can you see that the cells from the victim, I mean the band pattern of the victim, matches the band pattern of the female cells? These samples have been taken probably from two different locations and they match with each other because they come from the same individual. How about the sperm DNA which is found? This sperm DNA does not match with the sample of the boyfriend, but it does match with suspect 1. As I've already said in the beginning, that these cases are complicated. Sometimes there may be false allegations. Maybe the sexual intercourse was by consent and the female later on falsely accuses the suspect 1 to have raped her. There are many more such complications which may be involved. DNA fingerprinting is a very important and effective and quick tool to tell us where the sample is coming from, if at all we have suspects. If in case the victim gets murdered after sexual assault, then she has no longer lived to tell the tale, as it happened in the RG Kar rape case in Kolkata. So the victim died after that, and there was nobody to give out the names of the suspects. There were no suspects in that case, probably. So it gets more and more complicated because when there is a crime, there is someone thinking behind it. Obviously, they will try to save themselves. Any kind of crime is bad, but if at all the victim dies, it may not lead to any conclusion because no one can tell the story as vividly as the victim. Now let's talk about disputed paternity. We have DNA band pattern from mother, then from child, then we have three probable, we cannot call them suspects in this case, but three probable fathers because probably the mother may have had sexual intercourse with three of them when the child was conceived. The child matches with its mother in many of the bands. This band, this particular band. So many of the bands from the child's band pattern match with the same from the mother's band pattern. Now all you have to do is try to match with all three of them and see which one is found to be a better match. So I can see that the child has many fragments which are commonly shared with individual 1. Can you see this one, these two, then this one as well, the last fragment. At the same time, there are some bands which the child has in common, not many, but I think only this one band is common with the individual 2. Rest all the bands indicate that the individual 1 is the father of the child. So this is how DNA fingerprinting makes the job easy. So here in this case, it is individual 1, which is probably the father of the child. And in this case, it is suspect 1 who may have committed the crime. They will take more evidence into consideration. Many more things will be considered. Only then the law can take its course. Let's discuss one more case from the textbook itself. Here we have sample C coming right from the crime scene. There's a band pattern from DNA obtained from the crime scene. And we have two suspects, A and B. Here in this diagram, since we cannot show 26 fragments, they've shown only chromosome number 7, chromosome number 2, and 16. There are 2 fragments for each repetitive DNA because for every repetitive sequence, we will get the same from the dad as well as from the mom. So for every repetitive sequence, we have 2 fragments which we will obtain. Then all we have to do is run it through the gel and then obtain such band patterns. And can you tell me that the DNA from crime scene belongs to which of these 2 suspects? So this C is from the crime scene and it does match with the sample in column B. So that's how we can figure out that these 2 samples belong to the same person. If at all the individual B claims that I was not even there at that particular place, then how come his or her DNA is to be found from the crime scene? Well, again, like I've said, that evidence can also be planted. Sometimes it is the criminal which may have planted it. You never know. So let's not be a judge, but let's just learn from it that with this technique, you may or may not be able to find the culprit, but you will at least be able to match the crime scene evidence with the suspects. So finding the actual criminal is quite a complicated process and a difficult task. But this does prove to be an important tool in doing so. So now I will leave you with the 3 applications that are mentioned in the textbook. Let's discuss that. Well, DNA fingerprinting is an important technique which is used in forensics and it is used to solve problems of rape and complicated murder cases. They've already used the word complicated, which means that only by using this technique you cannot find the culprit. So many more things have to be taken into consideration at times. So next we can also find out biological father, mother, or sometimes both in case of disputed parenting. We can also employ the technique of DNA fingerprinting in pedigree analysis in cats, dogs, horses, and human beings. Because all we involve investigating here is polymorphism, right? And polymorphism is what we can use for pedigree analysis as well. So I leave you with this. Thanks a lot for attending. Take care.